So thank you everybody for being here. And I thought I'd start, because I think how many biologists were in this room, by telling you how we actually do biology, which might be a little bit different how you do mathematics. And the place where I will start is by showing you my lab. And so this is my lab. We usually have large groups, and mostly in biology, we do things in labs. It's experimental, but we depend strongly on bioinformatics, on mathematicians, and a lot of collaborators. So we really have, labs, and then also it costs a lot of money. So we're also, also fighting for money all the time. So we do have collaborations, and it's very interdisciplinary. And these are some of my collaborators. You will recognize there Maribel Hernandez, who is here with us, and she's really a pillar in the lab, because she is also, co-tutoring some of the students in some of the bioinformatics and modeling. And mostly all people here are from the area of physics and bioinformatics. And Joel Stevens is from Israel. He's my collaborator. He's amazing in physics, and it's great because we really need all these disciplines to ask more interesting questions and to find ways to solve them. And so it's highly collaborative. I also have a lot of experts in evolution and experts in ecology and microbiology, and we really need all this team to be able to do all these questions. So the work that I'll show you today has mostly been done with material that we get from a fantastic place, which is called Cuatro Ciénegas. And the geology has everything to do with the biology and the evolutionary story of the bacteria that I'll show you. And DNA is the basis of all this work. So most of the mathematics are mathematics, some that are done by the bioinformatics, and many with tools that are already available on the web, because everybody in biology is doing work with DNA. So you can see that for people working with humans, and to every other model, and also bacteria. I'll tell you what are microbial communities, to then, after explaining this great complexity of the community of 50,000 or more types of bacteria, to then tell you that we were able to have the smallest community, because our community has only 3 species and has all the complexity of a very highly diverse community. So I'll show you some of these. And so I'll start by taking you this script to Cuatro Ciénegas. So if you recognize this map, this is Mexico, and you find in the middle, Guanajuato, because that's where CINVESTAV is, where I work. Then you have Mexico City, with the southern. And then we usually have to drive 12 hours to go to this site, this field work. And that's Cuatro Ciénegas, Coahuila. And then you can even find Miami someplace. Can you? Okay, good. Okay, so where was all of this 100 million years ago? So it looks, like easy, that you think you'll recognize it, but you won't, because Coahuila had a shoreline. I mean, it was like Miami Beach, but now it's, after some time and all these tectonic movements and emerging mountains, now Cuatro Ciénegas, Coahuila, it's a desert, and this desert has no longer any of these, the beach is gone, but the water is there. Okay, so the water is still there, it's submerged, and it's fantastic because this water is generating a lot of ponds, and the ponds also have a lot of the microorganisms that lived there maybe 60 million years ago. So because now it's surrounded by very high mountains, you have what in biology we call a lot of endemisms. Endemisms are organisms that are unique of a place because they cannot exchange genes well, sex, basically, with other communities in other places. And so one fantastic thing about these ponds is that it has these beautiful stones that are not stones at all, are called stromatolites. And stromatolites are actually collections of microorganisms. These are communities of microorganisms that live together, and these are the oldest forms of communities, from the origin of life. So if you go back in time and ask how was the Earth, and the planet at the beginning, you will find that the planet, only has 5 billion years, a little bit more, and almost immediately you have you see these rocks that have evidence of the presence of bacteria. So these particular rocks that you see here are actually the stromatolites that I showed you before on the ponds. And although these are the history of life, the first evidence of living organisms here, we have the live bacteria in Quadrocerias. So we can actually go back in time and see how was this, the first bacteria that have these now signatures, in the rocks. And it took many, millions of years before this same bacteria changed the atmosphere of the Earth. So we used to have, a planet with no free oxygen, okay? And it took many years for bacteria like cyanobacteria to invent photosynthesis, and through photosynthesis to start liberating oxygen to the planet. So the planet changed from having bacteria that were actually killed by oxygen to evolving bacteria that would produce oxygen. And this is tricky, because some of the bacteria died because of oxygen. So this is, what the story of the planet. Story that starts with bacteria, and more than that, bacteria are forming communities. Bacteria never live by themselves. They always share resources and share a lot of things. And the most antique common community are communities that form mats. And mats are these kind of lasagna structures where you have on the top the green line, and then you have the purple, and you have the gray. And every of those layers, like this lasagna, has bacteria that are completely different and have completely different functions. And these functions have to communicate to each other what's happening, because the bacteria on top producing the sun have the photosynthesis, but the bacteria right below do not get any more oxygen, but they are still photosynthetic, another kind of photosynthesis. And then below, it's bacteria that actually die if they even see any oxygen. So all of these layers of biochemistry are communicating to each other to sustain life. And if you look at the biochemistry, if you remember your high school biochemistry with lots and lots of pathways, all of those pathways you can find them here, because bacteria actually invented all the biochemistry that we have nowadays in the planet. So if you see now this, in the clock, again on the life on Earth, what you see is that at the beginning of what was going on in the beginning, there was nothing. Then bacteria appeared, and then half of the time of the life of this planet, there was nothing of bacteria. Okay, so really the first plants, the first animals arrived or evolved very late. So most of the things that I told you, like biochemistry, it all comes from all these bacteria, because there was nothing else in the planet. So when everything else starts to emerge, like plants and animals, they immediately have to depend on the bacteria that were there. Now we have this microbiome, and then we all depend, like all the planet depends on bacteria. Even the oxygen of the planet, 20% comes from the ocean, from the same bacteria, from the same cyanobacteria. Okay, So I mean, it's not like okay, let's move on right now. Let's go for plants and animals. It's still an area that depends highly on bacteria. And if you think that this is like, oh yeah, these are ancient, microbial maps. I was there just 2 months ago with my team collecting samples, and we still find, the same maps. And of course, we bring them to the lab and work on them in the lab. So microbes are everywhere. And for all these years of evolution, we know that there are more than 50,000 species in every gram of soil. And that all of what's going on in the earth really depends on this, on bacteria. So there is a very young concept that you may hear about, which is something that people know as One Health. And One Health is about recognizing that bacteria are in soil, are picked up by plants, by animals, by humans, and that they are sustaining the health of the planet. Okay, the individual health of humans. And there are a lot of diseases related to dysbiosis and problems around our microbiome, and all the problems in agriculture also go around these bacteria that we are just not taking care properly of them. And there is therefore great interest in having mathematics for modeling how these communities still work in the planet, and how they will be affected, for instance, by climate change. So there is a lot to contribute to One Health by studying communities. And communities can be visualized as complex systems. Okay, given any change in the environment, they will have to adapt. Each of them, they are all interacting among them, and they are changing state. One important perturbation that you see in microorganisms are from antibiotics. Every time you have antibiotics, you are of course perturbing all your microbiota. But they really have every aspect that you see about in systems biology and complexity. You would find all these features in these complex communities. And this is how you plate bacteria that you take from the field. You take a petri dish and you sample whatever you have on the petri dish, and this is what you have. You have a diversity of bacteria growing there. And what we do in synthetic ecology, we want to reduce this complexity. We cannot work with 50,000 bacteria. We can tell who they are, but we cannot do experiments with 50,000, or at least not yet. Maybe we'll be one day like you, and we'll try to get them all together. And so these, what we do is to isolate them, go culture them, and then this is a bottom-up approach. Now that you have a lot of bacteria cultured, okay, one great thing about bacteria is you can freeze them. Okay, you can freeze them away, and then just take them out, and then so you can have the 50,000 bacteria, and then just take the one that you need every time. So this is what we want to understand in this bacteria. We want to do synthetic communities, that is artificial communities, where we can do experiments to understand resilience and to understand also things that have to do with complexity, such as special self-organization. But how can we even do that? I mean, if we have all these thousands of bacteria, what can we know who they are and what their function is? So I have to give you this little introduction to DNA, because everything we know about them is through their DNA. And in DNA, we have this code. It's a very simple molecule. This molecule is found in every thing that is living today, so that we have very amazing tools to get the DNA out and get the complete DNA sequence. And with the complete DNA sequence, I'll show you a little bit that you can tell who they are. You can tell their taxonomy. That's how taxonomy is done today. And then you can also have a lot of information about their evolution and how they are each related to each other. So just a reminder, this would be, sort of like what DNA would look like. You have a strand of DNA where those 4 bases are just forming combinations, and those combinations form genes. And you may, of course, you'll never find a DNA like this. I just made this one for you, where you will find, genes that you will be sort of like familiar with. You will remember your hemoglobin and your insulin and collagen. And for each one of those, you need to have a gene. And that's all encoded of course, in the case of humans, in 23 chromosomes with a lot of information. But the great thing is that once we do the biochemistry and have the genes, that is the sequence of, AGCT, then everything else we can do with bioinformatics. We can go to databases and ask what is encoded in this DNA that we sequence. And then everything else is really a deduction. We only know DNA, proteins, and everything is just a deduction, it's a hypothesis from the analysis in databases. So you have a very complex molecule, you have, just a lot of DNA, and you don't know what to do with that. But you have Maribel and have all the bioinformatics, and they will just, feed this information to the computer, and then immediately you have a lot of matrices that are going to be, done there to get something that looks like this. It looks like a tree. What this is doing is looking at the distance, comparing every strand of DNA in the database, and they will get the distance between different molecules to get what we call a tree. This tree is called a phylogeny. So what we have here is a piece of DNA from each of these verbs, okay? And these verbs, each one has the same sequence that you cannot tell anything about it. But when you arrange these molecules and do this distance analysis, then you can organize them in this kind of tree, okay? Where you can see which ones are closer together. So, like these ones in this branch, all these trees in the branch are represent like these not bacteria. I used, birds because I thought it would be easier than to see bacteria. We just do the same. So you have all these birds, and they are in the same branch, and they are very close together genetically. And then you also have other birds that start to get different, producing birds that are really different. Okay, so that's basically what you can do when you have DNA. You can organize that and get the distance between these sequences, how many mutations you will see, how many changes, and that tells you the evolutionary distance of them. And that's what we have done for all of everything that we know in life, create what is called the tree of life. And in this tree of life, what is amazing is that most of what you see are bacteria. So bacteria is mostly what dominates the planet. And we are here. See where we are? You follow the line until this green little thing. And that is where we are with the worms and other vertebrates. Okay? And everything else, on Earth is mostly bacteria. And another very important group of prokaryotes are the archaea. So I don't know if you know that archaea are very famous right now, and they've found amazing archaea in many places. And so basically, archaea, we're starting to collaborate with people doing that, but mostly in my work, we only look at this part of bacteria. So how do we even start to study these synthetic communities? So we have all these layers of experimental approaches to understand these communities. We start with the environmental sample. We do laboratory manipulations. We can study them in monocultures, that means one by one, like Aster did. Or we can start putting them together and make very small communities and see what happens if I combine strain A with strain B, and then you can do more complex consortia. But at all times, you will rely on information from DNA, from the genomes. This is what we know about the place that we sampled. So in Cienvegas, we have sampled, and we know, okay, because I cannot even take every these bacteria because they won't even grow on my petri dishes. So a lot of the information that we get is directly from soil. You basically take soil and get DNA out of there with alcohol, and you sequence that. You don't know what it is. You don't have them growing. Okay, it's called metagenome. So this is the diversity that we have in Quadriceras in just one single bond. And then among these bacteria, my favorite are these ones with the bacillus, and that's what we study in the lab. So what we study now is that we have them in monoculture, and now we do another particular way of sampling. Instead of sampling like everything, like a tube of soil, I just take micro samples because I want to understand what's going on in a very tiny community. So these tiny communities are only very few microliters, so that whatever I'm taking in those microliters are bacteria that are living together and are doing something together. So from these bacteria, what I want to understand is how did this community form? Why do we have this particular variety of bacteria there? Why do we have blue, green, and whatever color? What is going on there? Do they interact? And we know that in every community, bacteria are constantly interacting. And it could be that whatever we got out of the earth, it's just stochastic. It's randomly, it was there, it was born there, some things died, some things were born, and that's what we have. Or it could be that it's a deterministic model. It could be that they have particular features that allow them to communicate to each other, to interchange metabolites or nutrients, and they have to interchange these to live. But it could also be that some are killing each other, because bacteria are also amazing in the production of antibiotics. And they produce antibiotics because in these communities they have to fight for resources and they have to fight for space. So now you study one by one, and don't worry about all this information. What you see is that they are all different. Even if you take a few from a few millimeters of soil, what you see is that these bacteria are not doing the same. They have different features. Some, for instance, some can swim, some can do something that we call the biofilm, some can do other things like swarming, and they can do all kinds of tricks. But even though you think it's the same bacteria, like come on, it's all green bacteria or blue bacteria, they are not the same thing, they are different. In fact, if you sequence 3 of these bacteria that you think look exactly the same, and you get the whole sequence, and then what you see here is like their genes organized like one by one, all of their genes. What you find is that 30% of their genes are different. So I cannot imagine what each of you would look like if 30% of your genes were different among each other. And in bacteria, we think it's the same species, so it's the same bacteria, and it has 30% of their genes different. So there is a lot of room for combinations of communities that are making different things, and that's what we are seeing. So now what we do is, now that we have all these features, we ask something more interesting about these communities. If they are together, how come they don't kill each other? Or maybe they do kill each other. So what we do is we sample a few sites, and then each of these bacteria, we test them. And what you see here is that this bacteria that is showing like a ring. This is a bacteria that is growing on another layer of bacteria. And the ring is showing that nothing is growing there because everything there was killed. So we now see that bacteria are making antibiotics and are killing everything around them, right? They don't allow anything to grow there. Okay, so that allows us, yes? What is an antibiotic? Yes, it is different antibiotics, and I'll go to there. There, the bacteria invented the antibiotics. All the antibiotics that we use are just reformulations of those. And they can have at least 100 types of antibiotics, chemically different. It's a very complex. All of that is called secondary metabolite, which is things that bacteria don't really need. It's not essential for their lives, but they use it only to fight in communities. So if you organize bacteria and say, okay, who's killing who? Okay, who is doing what? So we have like the cartels here, okay? So we have cartel in cycle 2. In this cartel, we have on the top all these green bacteria that don't do anything. But then you have a bacteria like this square round of bacteria, and this one is capable of killing anything that is connected. So they really can, this other one is also connected and is killing a lot of bacteria. So you can have then 4 bacteria that are at the bottom that are killed by a lot of other bacteria. They're very sensitive to antibiotics. So what you could ask is like, well, if you have all these bacteria that have antibiotics that kill everybody, how come they are not the only ones in these communities, right? Because you guess that why they are killing everything else. So what you can do is use this data, and you can make an aggressivity index. You can see who is killing who, and with this aggressivity index, you can model this with a cell automaton and feed the data to this cell automaton and say, okay, this is the aggressivity index. I have this bacteria that can, of all 78, this one can kill 38. The next can kill 37. Some others can kill only 2, and some others are killed by 10. So you can feed all this data that is showing who is killing who and who is being killed. Okay, this is a very dramatic thing that happens all the time here. And this is what you do. You can feed the data and do a Petri dish, in silico Petri dish, and give very simple rules, which is if a bacteria is close to a not to one that you can kill, it's going to kill it and use that space. If it grows and moves to another square or there is a resistance, each one will remain, as such. So the question here is what is going on here? Can bacteria, get rid of the sensitive ones? And what happens is that there's this cell organization where you have patches where some of these bacteria get on. The sensitive bacteria are surrounded by resistant bacteria that do not kill them. So what you have here is that there is no extinction. And there is no dominance by bacteria, even though they are, real serial killers. So you find that there are these patches that are, created just locally by these particular interactions. If instead of allowing this structure, to be like that, you sort of like agitate your in vitro Petri dish and let them interact more, then you have more opportunity for the killer bacteria to find sensitive bacteria, and then those will go extinct. Those will be the ones that will immediately, find, would be away from this structure. And you will then end up only with the resistant and the antagonist bacteria. Okay, so we already have a consortia, we have a group of bacteria, and can we now make a synthetic community? Now we know who kills who, and maybe we can do something about it. So we have again our pyramid, and we also know taxonomically who they are. And we can then choose a strain that is resistant, a strain that is sensitive, and a strain that is antagonist, and see what happens in real lab experiments. But before that, we actually sequence the genome of all of these bacteria. And what we know is that every time we find a sensitive bacteria, they always belong to the same taxonomy group. If we get up to the antagonist bacteria, they always belong to the same taxonomy group, and the resistant bacteria too. So there's already something that we are learning about these communities, is that they are taxonomically related. So now we can take a bacteria that is resistant, an antagonist, and sensitive. And of course, we were careful to know that we could break them, and we have a different phenotype. What that means is that we can tell each other. We can know that the yellow is sensitive and the white is the aggressive, so we can tell them apart, even by the morphology of their colonies. So what we can do is to grow them separately. We can grow each of them in a different tube, and then we can put them together, and then take samples every time, every once in a while, like 5 minutes, 20 minutes, 30 minutes, and see what happens. So what happens when they interact, for some minutes? Just the two of them, like the antagonist and the sensitive. So the antagonist is going to be the one that you see like white, that is surrounding this. And among those antagonists, I mean, that's the killers, the yellow one cannot grow. Okay, you only can see growth of the white one, which is the killer. Okay, so only when there are a few opportunities to be away from those, you can grow. And we basically are seeing a cellular automaton in our plate. Those are the same results. We now see this patch of bacteria that are surrounding and protecting these other sensitive bacteria. But what if we do this in liquid and we take each one and see what happens, during time? So let's see if you can bear with me to see this particular line here, the green one that goes down. What this is happening is that when we put together the killer and the sensitive, in 5 minutes, 60% of the sensitive are dead. Okay, it's just going dramatically low. And then there's a little bit of maintenance there until the end. And then this would be the antagonist, the killer, and the killer can be there and nothing happens to it. Okay, the killer is very robust, and the sensitive goes down immediately. If we do another combination, if we have the resistant and we combine it with the sensitive, nothing happens. They are both, just maintained. If you also have the antagonist and the resistant, nothing happens. They are just remaining. So what happens if this one that goes down immediately and is killed by the antagonist, what happens if we now put 3 bacteria, if we add another one to the system? If we add the other bacteria to the system, the resistant, there's no more killing. In seconds, there is a change of state, and the sensitive is no longer sensitive, or I should say, I have no idea what's going on, or the killer is not killing, or the sensitive is resisting, or the resistant is, in the middle of the way and don't allow any of this. So this is basically an emergent property, okay? Where something that you predicted that to happen because of what you knew that the antagonist would kill the sensitive. Now that you have a third one, you have no idea what's going on. And this is the system that works in minutes. So this is the only system in biology where you can have only 3 bacteria, where you can have a response in 30 minutes, and where you can see, with the emergent properties with only 3 elements of this community. So we're producing some of the things that we have in very complex communities. And one of the things that I'll show you a little bit more, it has to do with antibiotics. So this is very important because every time that you get an antibiotic for something, maybe this is what's happening in your gut. And maybe this is what's happening with all the infections that we got. And it has to do with a lot of tolerance or resistance that happens immediately. Okay, well, maybe this one is a bit too much now, but what I'll just tell you is that if this thing that happens, that in 5 minutes, in 20 minutes, one of these bacteria dies, what happens if we get fresh bacteria into the same tube? If we get fresh bacteria of the sensitive into the same tube, now what happens is that it's not going to die. So the bacteria that killed the first batch of bacteria now got adjusted to the system. Now it understood that what it takes to be the winner, and it doesn't need to be aggressive anymore. So when you feed new sensitive bacteria, it doesn't do anything to it. And if we do the opposite, if we now take bacteria that was in contact with the antagonist, and then you take the survivors and grow them all night and feed them again to the antagonist, now it survives. So bacteria can change states, and you cannot predict what's going to happen. If you knew they were already, like you knew that this antagonist was an antagonist, but 5 minutes later is not an antagonist. The bacteria that was sensitive 30 minutes later is not sensitive anymore. So you really have a very complex system, even though it has only 3, that has all these properties. When you have the third bacteria, somehow it's in charge to have and immediately resolve the situation, and you get no killing. So, you have a lot of complexity here. And instead of a model that used to be very simple, which was this one, what we have now is that all these interactions that are changing the state of each other's bacteria interaction, so that you no longer can predict what's going to happen. So what if now we want to know what exactly is happening? Because we want to know what's happening. So now instead of going to DNA, we go to RNA. Okay. So RNA is this molecule that you can from DNA you get RNA, and that RNA is telling you this is what's going on in the cell, because this RNA will be turned into a protein. So if you get the RNA, it's telling you what's going on in what's the function of this bacteria. So what we do now is that we do the same by experiments, but now every so time, like maybe 5 minutes or 20 minutes, now we make, we produce RNA. And then what you want to do is see what are these bacteria doing when they encounter each other. What are the proteins that are made to survive this community antagonism? So what we do is we get all this RNA, and then bioinformatic people, take them apart and decide which RNA belongs to each of these bacteria, and then it tells you, this result. The results mean that green is the state of a gene before the interaction, and the orange is the state of the gene when it was in contact with the killer. Okay. Because what you want to know is, what do you do if you find a killer? If someone enters over here, I mean, you certainly do something, and that's what we want to know. What do they do? And what we find is that immediately the action is to get out a lot of defense mechanisms against antibiotics. Those same defense mechanisms that make our bacteria multi-resistant in hospitals, those are exactly the enzymes that are turned on to contend with the killer. And so you have all the enzymes that also contend with the stress, just oxidative stress, like a catalyst. And you have enzymes like this one, which is called POSB. That enzyme actually breaks off an antibiotic. The antibiotic is there, and it just breaks it out. So we really understand what's going on. We can use databases that are specialized in understanding what each gene is in these things. And this is what Ernesto was asking, what kinds of antibiotics are there? Everything that you see on the top are the types of antibiotics that have been gathered for all bacteria that we know. And here we have the strains. And we have that all the antagonist bacteria make the same kind of antibiotics. All the sensitive bacteria make few antibiotics, but they make a few things. And the resistant bacteria make a lot of antibiotics too. So all of them are fully loaded with armament to contend in these communities, and they just deploy this at different times. Now what we want to do is to see, can we, now we have used only 3 bacteria and 3 very specific bacteria. And I know the taxonomy group, I know that the resistance are all blue, okay? They have very interesting names that you want to know. And then you have the antagonist bacteria, and we have a lot of antagonist bacteria, and you have a lot of the sensitives. So can we do combinations and get the same result? Can we get these emergent properties, and this one dies, and this one doesn't die, and then there's this emergent property? And the result is no. No, it's I don't know, the just good luck of my students to get this first, 3 strain, because it's very difficult to get to reproduce results when you exchange bacteria. Okay, so even though they look very much the same and the sequences look the same, there are differences. So how did I even start this project? And this is what I proposed to colleagues, and I really proposed this. And I wanted to know, how did Mike Tyson, make his plans for combat? And Mike Tyson, once said that even if you have a plan, you will get punched in the mouth. Okay, so it's like everybody has a plan. But I said, bacteria must have a plan. They've been in the planet so long and rehearsing this in communities. So they must have a plan. And we find that the plan is encoded. We can decode the plan, and they do get punched, but they are many. They are populations, so you have basically survival through their population. Okay.
Thank you. Fascinating questions.
I'm interested in the community that you discussed at the beginning. So it's a community of the same species?
The one that is in many layers?
The one that looks like stone.
Oh no, those are an amazing diversity, many species.
But every layer is a different species?
Every layer is many different species. Yeah.
So why different species? How different species decide to make a community?
So we think there's a lot of redundancy, because if you take one of these maps from Australia, because in Australia there are also some of these maps, you may have different groups of different species, but the same functions. So you can find the same functions even in different species. But yes, every layer you can have maybe 100 different species.
It's a master plan for many species.
Yes.
It's very unusual.
Exactly. There's a lot of redundancy.
Trade-off. There is no genetic trade-off.
Other questions? One, some years ago, I wrote a paper about what I ended up calling competition-mediated coexistence, where this was 3 species, and I did this sort of at an ODE, wow. And then I did it also with where you had randomly moving kinds of things. And okay. What the thing was that you had to go to like added higher order interactions, where the idea here is then I could get the phenomena where you could have a species enter that either one of 2 things could kill, but when you had them all together, all 3 could coexist. So we'll talk.
Wow, you just did this, did the modeling, and now I'm confirming your data. Yeah. Okay, sure. Thank you.
Any other questions? Gabriela.